I'm Bonface a PhD student at the Wellcome Sanger Institute. We stain bacteria to make them visible. By using a specific staining technique called gram stain, the dyes themselves will also be a crucial tool to identifying them. Now I'm going to show you how to do it. Step 1. Preparing a smear slide. It is crucial to do this in an aseptic or sterile environment. So, I'll be working near this heating block. The heat creates an air current that helps to keep contaminants away from the workstation. You can also create an aseptic work environment with a Bunsen burner. Label the slide with your name, the date, and the sample details. Using a sterile inoculation loop, put 1 drop of water on the center of the slide. Next, lift the lid of the petri dish just enough that the inoculation loop will fit through. Collect a small sample from your isolated bacterial colony. Replace the lid straight away. Mix the bacteria with the drop of water. Be sure to mix the smear evenly and keep it nice and central on the slide. Leave the slide to air dry completely. Once the slide is dry, we'll heat fix it by placing it directly on the heating block. If you're using a Bunsen burner, quickly pass the slide smear side up through the flame 3 times. Don't hover the slide in the flame as this could overheat it. Step 2. Gram staining. We'll apply 4 different solutions to the smear in sequence. First, douse the smear with crystal violet and let it sit for 60 seconds. Then, use tongs to hold the slide at an angle and gently rinse it with distilled water until the water runs clear off the slide. Next, apply Gram's iodine. This locks the violet stain into the cells. Let it sit for 60 seconds. Rinse again with distilled water. Now, here is a crucial step that makes gram staining special. We'll apply ethanol over the smear 1 drop at a time. This removes the violet stain from some cells, but not all of them. This is how we can differentiate between gram-positive bacteria, which will still hold the violet stain, and gram-negative bacteria, which will not. Stop applying as soon as it runs clear off the slide. Then rinse again with distilled water. Finally, we'll add a counterstain so that the gram-negative bacteria are also dyed. For this, we use safranin, which is pink in color. Let it sit for 45 seconds. Then, rinse gently with distilled water. Again, stopping as soon as the water runs clear. Tap off any excess water and let the slide air dry completely. Once dry, we'll use a mountain medium, which is like a glue, to affix a cover slip to the slide. Now it's ready to examine under the microscope. The gram-positive bacteria will be purple and the gram-negative bacteria will be pink, easy to see and identify. That's it. Now you know the techniques behind making a bacterial smear on a slide and gram staining, 2 fundamental techniques in microbiology.